Good afternoon, everybody, and thank you to OIS for allowing us to present today. First of all, Azura's mission is to create a whole new class of ophthalmic keratolytics by really focusing on the skin and the glandular structure that supports the ocular surface. And in doing so, we're really targeting the root cause of a lot of the ocular surface manifestations. Our lead program, AZR-MD001, is focused on meibomian gland dysfunction. To this audience, I don't really need to explain meibomian gland dysfunction, but I just want to highlight it. From our perspective, it is actually the #1 cause of a lot of downstream effects. Tear film changes, tear film instability, which then ultimately results in dry eye disease, other issues around visual quality, as well as one of the key causes of contact lens discomfort and intolerance. And if you don't address these glandular changes, it can actually lead to ultimately changes in glandular function, as well as potential for glandular atrophy and other issues, which ultimately results in one of the focuses of other areas is ocular surface inflammation. We all know it's a very large unmet medical need. Our lead program is a keratolytic. It's selenium sulfide with a unique mechanism of action. It's a keratostatic, so what that means is it actually slows down the cell replication cycle, in particular the keratinocytes at the gland orifice, and therefore reducing the production of keratin proteins. It's a very potent keratolytic, so it's not an anti-inflammatory. It focuses on breaking the disulfide bonds that form due to the changes in the local antioxidant environment, which causes protein aggregation in the meibum. It has a unique property that was demonstrated in its medicated shampoo function, where it actually increases secretions from the sebaceous glands. So MGD being a disease of poor quality secretions, as well as poor quantity secretions, having something that can unblock the glands, improve the quality of the meibum, and potentially increase secretions is a unique opportunity. In November, we released the results of our first registration study. It was a 3-month primary endpoint with a focus on 2 co-primaries, how many glands are actually open, so meibomian glands yielding liquid secretion, as well as ocular surface disease index. One thing to note about our formulation, it's not an eye drop, it's an ointment, and it's actually applied to the lower lid margin 2 times per week at bedtime. So something very, very different. So let's look at the 2 co-primary endpoints. In this slide, you'll see the meibomian glands yielding liquid secretion, the change from baseline compared to vehicle. And as you can see at day 90, a very statistically significant result. What's really impressive, if you look at day 14, so with only 4 applications of drug, we see a doubling of score. And so that's one thing that seems to be very unique about this approach. We do see a very fast response. In terms of the improvement in symptoms, we saw a statistically significant improvement compared to vehicle of the 0.5% compared to vehicle. One of the things that we really wanted to understand was what were the clinically relevant endpoints that matter to clinicians. So we had a number of pre-specified secondary endpoints. If you look at the literature, a change of 5 glands of number of glands that are open, it strongly correlates with symptom improvement. And by day 90, we see 45% of the patient population in this study achieving more than 5 glands that are open. TFOS defined meibomian gland dysfunction as anybody who has a meibomian gland score of less than 12. So that's a measure of the number of glands that are open multiplied by a quality measure on a 0 to 45 scale. So we use that as a cutoff. And as you can see, by day 90, almost 68 or 70% of patients actually achieve that level. So by definition of TFOS, they don't have meibomian gland dysfunction anymore. Once you open the glands, improve the quality of the meibum, the next expectation is that you'd increase tear film stability. And as you can see here with the measure of tear breakup time, and we use low fluorescein volume in this study, and as you can see, a significant improvement both from baseline as well as compared to vehicle in tear breakup time. Ultimately, we want to make sure that patients decrease their symptoms, and we use the ocular surface disease index because it's the only validated endpoint that has cutoffs for disease severity. And so anybody who achieves a score of less than 13 is considered asymptomatic. And as you can see, we achieved almost 50% of the patient population becoming asymptomatic in this study. We also looked at a number of other patient-reported outcomes, and just due to time, I can't share all that with you, but one of the ones that we looked at was speed, and as you can see, again, a statistically significant improvement versus vehicle and compared to baseline. One of the interesting things about our approach is that we're focused on the glandular morphology. And so what I want to share with you here today is really some of the other elements of what patients feel around ocular discomfort. And as you can see, overall, under the visual analog scale, an improvement compared to baseline. In terms of adverse event and tolerability, it's a keratolytic. So we do see some burning and tingling on application, so that's on the eyelid, but it was only observed for the first 5 to 10 minutes, just before bedtime, and it decreased over the first 4 to 6 months. What was really interesting about our study was that 62% of the patients actually came into the study having level 1 or level 2 Oxford staining, and we didn't see an increase in that patient population. All the treatment adverse events were mild to moderate. We only saw across the whole study 3 patients drop out of the study due to adverse events. So in summary, we hit our co-primary endpoints from a regulatory perspective. We have significant clinically relevant performance where 46% of patients actually have their glands opening to a normal level. What's really important in terms of their overall quality of meibum, 68% of patients actually achieve normal meibum levels. But what's really important is that actually patients begin to feel better, particularly around ocular surface symptoms. So with that, thank you very much.